second speaker of the day, and as usual during CORC, we do again one speaker is outside CORC, and then one is within the CO2 Research Center. And today we have the pleasure to host nothing but the director of the center, Professor Alfred Spormann from Stanford University. Alfred is a professor of both civil and environmental engineering and of chemical engineering, and he's been working at Stanford for the past 2 decades on especially microbial processes that are involved in a variety of different transformations, including wastewater purification and chemical reactions that can use small building blocks and convert it into more important and valuable chemicals. And today's topic of Alfred's presentation is, in fact, the use of the engineering of microbial metabolism for energy and environmental applications, and in particular, in the conversion of CO2 to other small molecules. Thank you so much, Alfred, for joining us today. And please, you can share your slides, and the floor is yours.
Thanks, Matteo, for the kind introduction. All right, can you see my slide?
Perfect, yes.
What I want to do is actually to use the next 20 minutes and give you a microbial perspective of what we believe is a key platform for CO2 utilization for storage and for conversion, and that's microbial electrosynthesis. I think most of you remember from Jörg's talk last week what the electrosynthesis is, but briefly, we are using microbes to essentially convert electricity and CO2 into interesting compounds, C1, C2, C4, and up to C6 or C8 compounds, which can be used and the CO2 can be released, or as we just heard with Matteo, if, for example, this methane then can be used in pyrolysis to make carbonaceous material, so that in the end, the climate active CO2 carbon is trapped as an inert carbon. And what is really attractive on this microbial electrosynthesis platform is that this is mediated by microbes, anaerobic microbes that you find in many ecosystems. You don't need to engineer the organisms. And they catalyze these reactions at high rate and high selectivity at room temperatures and no pressure. And that's something that is clearly interesting, where this microbial process then can be also placed in the context of the chemical process, as what Matteo just described for pyrolysis and to make interesting carbon materials. And in the end, we would make this from CO2. So I want to talk a little bit about the microbial aspect. What are the parameters that are important for microbes to catalyze this microbial electrosynthesis? And I should say, the input here is electricity from typically renewables, solar, wind, but you can also think of nuclear energy, and the electrons for the CO2 reduction, and it comes ultimately from water, or it can also come from waste. We and others worked on this over the last almost decade, and this is just a schematic representation of how the system looks like. In reality, a lot has changed, and these reactors look a little bit different, but you have a cathode here with a certain biocompatible material suspended in a microbial reactor, where you have microbes that use ultimately hydrogen that is produced in situ on these biocompatible electrodes and metabolize hydrogen to compounds with plus CO2 to, for example, methane or acetate or ethanol and so on and so forth. In order to have this hydrogen production here at the cathode, we have an electric current from an anode here, which oxidizes water. We have a proton exchange membrane here that separates the anode from the cathode compartment, and also keeps oxygen out, because these processes are anoxic, meaning that they only occur in these microbes when oxygen is absent. So with this process, you can basically feed the organisms that naturally would use hydrogen, electricity, and CO2 and make interesting compounds from that. And just here in the corner, you see actually how the system looks in the lab on the research scale. We have a temperature control here, and here we have a cathode, anode compartment, and then a cathode compartment here. So now that looks great, but the question is, if we want to use this microbial electrosynthesis process at scale, it has to work reliably well. So there is a question about how robust can biology deliver actually this really interesting task, and what are the limitations and benefits for that. So we focused on one aspect which was the dealing with intermittency. Actually, I should go back. This process here of electrosynthesis, there are two important aspects of that. On one hand, you can say store electrical energy here. So this is electricity in form of chemical energy. That's an extremely interesting aspect for storing electrical energy in form of as a relatively stable molecule. So you can store this energy over longer periods of time. That's basically natural gas, renewable natural gas. Or you can look at this technology platform to convert CO2 into something useful. So it's a carbon CO2 technology, but also an energy saving technology. So we looked then in more detail to what extent can microbial electrosynthesis store intermittent electricity. And here you have a profile of light energy. As the sun rises, there's energy available. This energy is not always used when it's actually produced. For example, here are the peaks when energy is most used. So a lot of energy from photovoltaics, for example, is curtailed because there is no use for that. So the question then is, what about if we can use this curtailed energy and produce methane? So we save energy, we save actually a lot of money, and we are producing a fuel which is carbon neutral and then can be stored in the natural gas grid or used and other times. This graph shows that the microbes actually can do this very well, and I think Jörg showed this last week. Here we have the simulated over a period of 1 month. So this is an experiment that goes over 1 month. The simulated energies that are typically curtailed, and we are feeding them as current into the microbial electrosynthesis. So you see that the periodicity is right. The intensity is different because, well, there are cloudy days and there are sunny days, and therefore you have a different electricities. We then plotted here the amount of methane that is formed, and you can see here that this amount of methane formation very well and nicely mirrors the available electricity. So that's a great match. We also see that the amount of hydrogen that is unused is very minimal here. So you have a few spikes here and a few spikes here, so that the overall Coulombic efficiency is between 90 and 95%, as you can see here. If you use a little trick here, and it goes back to the question which I want to really address, is how do cells actually deal with this intermittency here when there is sort of this period between feast and famine. There's energy, electricity available, and they use hydrogen. And then there's the night, basically 12 hours when there is starvation. How do the cells really deal with that? And how can we use these properties in the microorganisms ultimately that this can work at scale? In this part of the experiment here, we simply applied a little background current, which was like 0.5%, which was constant, overlaid over these fluctuating energies. And as you can see here, where you compare actually this graph to this graph here, there is almost a neglectable amount of hydrogen produced. Just forget this here. This is where Sturbar got stuck. And you can see that the efficiency here, the Coulombic efficiency, is really stable, or really very high. In other words, if you give the cells a little sandwich, in other words, a constant flux of really, really low energy, the organisms much better deal with sudden increases in available energy or hydrogen than if they are completely starving. So the question is, what makes these organisms so responsive? And this goes back to what is on the more fundamental side, a proteome allocation model. So if you think of a microbial cell here, this is sort of the outline of a microbial cell. There are proteins in there. And I just want to show you here how you can think about how crowded that cell space is in a microbe. So this is a simulation here where you simulate 130 nanoseconds. So it's much shorter than the blink of an eye, the movement of macromolecules. And you can see, first of all, the cell is really packed. So it's not just a solution or a dilute solution, but it's really packed, where you have a lot of contact between proteins, between ribosomes, between RNAs, and so on and so forth. So in other words, what kind of proteins with what functions a cell puts inside is actually very limited, and there needs to be sort of selected for how this can be achieved. In this simulation here, you're looking actually at metabolites, small molecules, that are produced by bacteria. And you can see here, they can diffuse quite a bit where there is a space, but between macromolecules, there is only sort of limited movement. So the point being here, it really matters how the cells divides up this available space for proteins that have certain functions. One important function is the catabolic proteome. So these are all these enzymes involved in energy conservation that powers the cell. The second aspect is an anabolic proteome. So all these proteins are involved in biosynthesis of cell components for a growing cell. And then an anabolic proteome, which is primarily the ribosome. So you can count the ribosomes also here, but ribosomes play a significantly, so like this here, play a significantly different role and require a lot of space. We know from E. coli that when cells grow slowly or fast, the allocation of this proteome is different. And this becomes relevant for us because in the periods between feast and famine, so between light, available energy, so energy is available, and darkness with no energy, we are essentially cycling the cells between these two states. E. coli, as the quote-unquote standard model organism, when it grows aerobically on glucose, changes the catabolic outfit to adapt to the flux of nutrients under those conditions. So when the cells grow slower, they have actually a larger catabolic proteome than when they're growing fast. And they have lower number of ribosomes because the rate of protein synthesis is actually lower than when they grow fast. So in other words, E. coli adapts genetically in terms of changing the proteome composition largely sort of over several important sectors in this transition. The question is, and I'm moving here forward, how do the methanogens that produce methane from CO2 and electricity do that? So the work here that I'm talking is by Albert Müller and Wen-Yu Gu, two postdocs in the lab, who looked at the proteome allocation in chemostat when we can control the growth rate of the methanogen under specific conditions. And we measured every parameter, more or less, that you can measure in order to characterize and understand what makes these methanogens so stable and so responsive to intermittent energy. So we changed the dilution rate, and means the growth rate, where in the end, the growth rate of the organisms in a chemostat range between 13 days. So the doubling of a cell takes 13 days, or on the fast side here within 3 hours, and then measured proteome composition, cell size, ribosome content, ribosome activity, and many other parameters. And I just want to focus here on two important aspects. So here we are varying the growth rate, and again, this is basically the cycling between almost no growth and fast growth. And we want to see what the spectrum and the biology is of how cells adapt to that. So when the cells are limited by formate, which is a soluble hydrogen equivalent that we used for simply experimental reasons, you see that the fraction of proteins that make up the enzyme, the pathway for methanogenesis, is actually constant. So in other words, although the cells, when they grow slower, produce methane much slower, they still have the same number of proteins and the same fraction of proteins encoding for methanogenesis. When the cells are limited by phosphate rather than by the metabolizable formate, it is also constant, but it's actually at a slightly lower level. So that's different from E. coli, because we know E. coli changes with the growth rate, the catabolic proteome. So the catabolic proteome here stays constant. In other words, the cell have a constant level of proteins that allows for methanogenesis. And that's unexpected. That's against the E. coli paradigm. And it is very useful for the electrosynthesis. Let me just move this out of the way here. Actually, let me skip through that. That basically says the same thing. Okay. So this is actually probably the most important slide here, and I'll walk you through this. That explains, and I'll give you the punchline right here, that explains why methanogens are great organisms to use for intermittency, for long-term stable conversion of hydrogen and electricity into methane. Here's again the growth rate that we plot, slow growth, or here almost no growth, and here's fast growth. And here, the dotted line with the black line is essentially the rate of methane formation that the cells have as they grow in the chemostat. So as you would expect, when the cells start to grow slower, the rate of methane formation decreases. On the other hand, I showed you that the level of proteins for methanogenesis actually stays constant. That means the cells should have the capacity for high rates of methanogenesis, although they're growing at a slower rate. I hope that makes sense. In other words, the cells seem to have the pathway for methanogenesis ready, although they don't know this. So in order to test that, we took cells from the chemostat grown at different growth rates, where we knew the proteome composition, and then tested these cells for maximum rate of methanogenesis. So we took them out of the constraints to grow slow, just put them into a new container with unlimited resources and asked the question, how fast can they produce methane? And these are actually here, these gray lines. And what you can see here, for most of the dilutions, when you take the cells out of the chemostat and test what is the capacity for methane formation, that is actually almost invariant, although they grow slower. There's an interesting aspect here in the very slow growth, where we do have still all proteins for methanogenesis present, but the rate in vivo that we see actually is lower. So that basically explains why methanogens are great to respond to intermittency, because they do not change the pathway enzymes, in layman's terms, the pathway enzymes for methanogenesis, which basically keeps, so to speak, the spring loaded to respond to increases in substrate concentrations with a high rate. I don't know if this makes sense, but that is so fundamentally different from what E. coli does. The cells have a constant rate, a constant level of methanogenesis proteins. That is actually different under anabolic limitations. So under anabolic limitations, when we limit the cells for phosphate, you can see that the specific rate of methane formation, when you take the cells out and give them unlimited resources, actually decreases. So that means, from an operation point of view, you want to make sure that cells are limited by hydrogen or formate in this case, and not limited by phosphate, for example. I think I want to skip through this here and just summarize that the fundamental difference here between the paradigm of E. coli and Methanococcus maripaludis, so the methanogen, is really that the methanogen does not change the proteome composition. Ribosome levels also stays constant. It's a completely different microbial strategy to persist in an environment. And this probably has the advantage to rapidly respond to differences in metabolic flux, and that's what we see with the intermittency. Whereas E. coli, which is an enteric organism, changes the entire outfit and proteome composition and pathway composition, also changes the ribosome content when it goes from slow growth to fast growth. This is all constant here in methanogens. So I think this actually provides the basis or an explanation for why methanogens actually are really good in making methane and using all resources up, and can respond to fluctuations in intermittency. I think in the interest of time, I'll probably skip through this. This basically says that the proteome is also pretty stable, regardless if you grow the cells with hydrogen that is formed electrochemically in situ, or if you just feed hydrogen and CO2 gas into a fermenter. That's something that we expect. But the important aspect here is that the cells in this electrochemical system are also subjected to electric field. And since the composition of the proteome is almost indistinguishable from when the cells just go on hydrogen that is pumped in without electric field, it tells us that the electrical setup actually does not limit the microbial metabolism. So these are good news. And this is sort of a recent picture of the group that has been doing the work. Not in the picture is Albert, who actually was chasing his kids. This is when you, and yeah, I think I want to stop here, and I'm happy to take questions.